Good morning, everyone. As Jaap told you, my name is Frédéric Jehl. I am a first-year PhD student in the team Genetics and Genomics of the joint unit INRA Gros Campus Ouest called Pégase, and I'm located in Rennes in France. And this morning I'm going to present you some results of our work on the layer's response to a suboptimal diet through phenotypic and transcriptomic changes in 4 tissues. So let us begin with a few words on the context that motivated this study as well as its objectives. So for the context, we are at the moment in a situation where few main important breeding companies are responsible for the selection of the layers used by the worldwide egg production sector. This means that layers that were selected in a controlled environment are exposed during their production career are exposed to environments that can be more or less challenging because of stressors such as high or low temperatures, or to variation in their diet composition or their diet energy content, which is what we study here. So this study had 3 main objectives. The first one was to study the impact of an energy-depleted diet, which was a diet containing 15% less energy than a commercial diet, on the performances of layers. Our second objective was to explore the molecular mechanisms at work at the gene expression level in 4 energy-related tissues of these layers. And finally, our third objective was to study or to investigate a potential interaction effect between the low-energy diet and the bird's feed efficiency by using 2 lines diverging on this character, yes, called the RFI plus and RFI minus lines. And we studied feed efficiency because, as you probably all know, it is a trait of great economical importance. So to do so, 50 birds of each line were fed either a low-energy diet that contained 15% less energy than the control diet, or the control diet, during 14 weeks. During this period of time, we had several measurements were realized on these birds, such as body weight. We measured their feed intake. We measured some parameters linked to the eggs, so the egg number, egg weight, yolk weight, and shell stiffness. And at the end of the stress period, the birds were slaughtered, and we weighted their abdominal adipose tissue. In addition to these measurements, at the end of the stress period, we selected 32 animals, that's 8 animals per group, in order to realize a transcriptomic analysis. And we realized this analysis on 4 tissues that we sampled and that are all involved in the energy homeostasis. These tissues were the adipose tissue, the blood, the hypothalamus, and the liver. So we use this tissue to realize the RNA-seq analysis, the steps of which I briefly outline here. So we did at the lab bench the RNA extraction and the RNA sequencing. And then for the bioinformatics steps, the sequence mapping on a reference genome, an expression evaluation, followed by the differential expression analysis and the pathway enrichment analysis. Basically, this kind of analysis of differential expression analysis aims at answering two big questions. The first question is, in each tissue of interest, in each tissue that we studied, which genes are differentially expressed, that is, have an expression that is not the same between the 2 lines or the 2 diets, or are under an interaction effect. And the second question that is answered by the pathway enrichment analysis is, in each of these tissues, which pathways, so which biological functions are affected in the light of these differentially expressed genes. So I'm now switching to the results. So our first result here is that the low-energy diet had no effect on production. As you can see here, neither the egg number nor the egg weight were significantly different between the birds from the low-energy diet group compared to the control group. And our interpretation of this result is that the layers adapted to this low-energy diet, at least from the point of view of a productive point of view. However, this adaptation was done at some cost. The first one is, as you can see here, the feed intake of the birds from low-energy diet group was significantly higher than the one of the control group. And at the same time, both the abdominal adipose weight and the yolk weight of the control group was superior to the one of the low energy diet group. So to summarize this slide, we can say that the low energy diet led to an increase in feed intake and to a diminution of fat storage. So this leads us to the first set of conclusions. So low energy diet did not affect layers from the point of view of production, and the low energy from the diet was compensated by an increase in feed intake and a diminution of fat storage in the eggs, as we've seen by the lower yolk weight, and in the body, as we've seen with the lower abdominal adipose weight. Our second conclusion is that we found no interaction between the line and the diet. As you can see here in the rightmost column, none of the traits that we were interested in had a significant interaction, which means that basically the birds reacted to the low energy diet in the same way, regardless of their line. And that's why we focus on the diet, and that's why I presented you only with result linked with the diet. So now, having seen that, a question that arises is, what are the tissues that were affected by the low energy diet? And in these tissues, what are the genes that underlie the adaptation to the low energy diet? And that's the question we answered using the transcriptomic analysis. So here, our first result concerning the transcriptomic analysis is that the low energy diet had a contrasted effect on the tissue transcriptomes. As you can see here, among the almost 25,000 known genes in chicken, only 2 and 15, respectively, were differentially expressed in the adipose tissue and in the liver. On the other hand, we found more than 1,000 genes differentially expressed in the blood, and almost 3,000 genes differentially expressed in the hypothalamus. So we conclude from this observation that the low energy diet had almost or had no effect on the adipose tissue and on the liver, but affected the blood and the hypothalamus transcriptomes. The other conclusion that we can draw from this slide is, as you can see in this last line, we found almost no gene that were under an interaction effect in none of this tissue, which means that the genes reacted in the same way to the low energy diet, regardless of the line of the birds. And these results, of course, strengthen our previous results of an absence of interaction that we observed at the trait level. So now I'm going to focus on the hypothalamus in order to present you or to highlight one of the mechanisms that we propose, that we found, that we may have found in the hypothalamus in order to explain the increase in feed intake. So what is this mechanism? Studying the genes of the differentially expressed genes in the hypothalamus of the low energy diet group, we found that genes related to the omega-6 polyunsaturated fatty acids, or PUFA, biosynthetic pathway, were overexpressed in the hypothalamus of the birds from the low energy diet group. These genes are the FADS2, ELOVL5, and FADS1. The omega-6 PUFA biosynthetic pathway uses fatty acids from the diet in order to synthesize in chain different omega-6 fatty acids, one of the most important of which is the arachidonic acid, or the SYN24 omega-6, which has a wide diversity of roles in the metabolism and in the body. And interestingly, we found that among these roles, the arachidonic acid can be added on phosphatidyletanolamine, which is a molecule, to form a NAPE. And NAPEs, through the action of an enzyme called NAPE-PLD, can form an andamide. This enzyme, NAPE-PLD, as you can see here, is also overexpressed in the hypothalamus of the birds from the low energy diet group. An andamide is a molecule that belongs to a family called the endocannabinoids, which is a family of molecules that have the particularity to activate a receptor that is the receptor that is activated by the bioactive substance of the cannabis. This family of molecules are synthesized by the body, thus the name endocannabinoids, and are involved in a wide diversity of mechanisms in the brain in general, such as pain modulation or synaptic development, or, most interestingly in our case, food intake regulation. And the gene coding for the receptor of the endocannabinoids, which is called CB1R, was found to be underexpressed in the hypothalamus of the low-energy diet group. And our conclusion here is that the combined action of the anandamide and CB1R leads or can explain in part the increase of food intake in the birds of the low energy diet group. As you can see here, these results are in line with what you can find in the literature. So to finish this presentation, a few take-home messages. So the low energy diet, as we've seen, did not affect the production of the layers, but increased the feed intake and lowered their fat storage, and did not affect, when we consider the tissues, did not affect the liver nor the adipose tissue transcriptome. And going further, we've seen that the increase in feed intake was likely mediated by an alteration of the endocannabinoids pathway in the hypothalamus. So to finish this presentation, I would like to thank all the persons who made this work possible, as well as my two PhD supervisors, Tatiana Jehl in Paris and Sandrine Lagaring in Rennes. And I would like to thank you for your attention. Thank you.
Thank you, Frédéric, for the nice presentation. Questions about energy diets and regulation of feed intake? Not yet? Frédéric, the fact that the animals produce the same quantity of eggs, would that mean that egg production, well, for the chicken, basically, the desire to reproduce, even though she's not reproducing, but the egg was there to reproduce, is that driving feed intake? And that is a priority because they maintained egg production, and the consequence is, well, then you have to eat more or mobilize fat.
Well, I don't know if it's really the need or the biological function of reproduction, because these layers were, of course selected through the years for the egg production. So I'm not sure about the reproductive side, but indeed it seems that this selection made it so that the production of eggs is more or less an above all mechanism in production in chickens.
Thank you for this presentation. I did not understand how the lipid content of the egg was measured. Because you said there is no, because egg is not a fat storage, it is fat exportation. And fat storage in the tissue, but in the layer, the main lipid metabolism is to export into the egg, and then it depends how you quantify all the lipids in the yolk.
Yeah, so about the measurement, I'm unsure of this, the answer. It was done by, obviously breaking the eggs, and I guess weighting the yolk. And for the other part of your question, yeah, indeed, we saw, or we conceptualize that the yolk in the egg was a form of fat storage, not for the chicken, the chicken who laid the eggs, but rather in a way for the chicken that could hatch from the egg. So, yeah.
Okay, let's thank Frédéric again for the presentation.
Thank you.